This lecture is a lecture for the Canadian Bioinformatics Workshop. It is freely available to share and distribute according to the Creative Commons license, as you can see here. Module 3, Network Visualization and Analysis. We are going to go through now. I'm hoping that by the end of this lecture, you'll understand the advantages of using network visualization. We're going to go over the introduction of what networks are and where we see them. We'll go over basic terminology of networks. We'll briefly look at some of the different network visualizations that there are, as well as different analysis techniques that we can use with networks to more easily describe and draw conclusions from our data. I'm not sure if you've heard, and you see, I could say this probably a little bit more easily now, right? 6 degrees of separation. What does 6 degrees of separation imply? It means everyone in the world is connected by at most 6 links or 6 degrees. Now, this is probably a concept that's a lot more prevalent nowadays because a lot of people are on social networks and understand that, people are connected to each other by a lot fewer steps than one would have thought. And also nowadays people are more connected just because you're able to communicate with people all over the world and this person's connected to that person. The whole idea of this concept was actually originally developed or demonstrated by Stanley Milgram's lab in Harvard in the 1960s. He did a very interesting experiment where he actually had, old school snail mail, had people mail out letters to 6 people that they know at random. And he asked that you put your name and some defining, I think it was maybe your name and your address on the list. And when you got the letter, you send it to 6 other people. Now, if you got to a point where you knew somebody on the list of people, you were instructed to send that letter back to the Stanley Milgram lab at Harvard. And what they uncovered that was most people, 2 different people, were connected by a distance of 6 degrees, right? So most people were connected by the 6 degrees of separation. This concept was further popularized in the 1990s with a popular movie game, which basically said that you can, 6 degrees of Kevin Bacon, right? You could get to any movie that was had Kevin Bacon as an actor within 6 steps. I guess he was a very popular actor in the 90s, in the 80s and the 90s. I'm not sure who you would say 6 degrees of nowadays, but I mean, people are connected in our social structures pervasively. So networks are everywhere, right? The example I've given here is the social network, but they're not just in the social network. They also exist within our power grids, our cell tower networks, our FedEx delivery networks, our supply chain networks. That was something that was very much on display during COVID, for instance, right? When elements of our supply chain network broke down and all of a sudden, people were scrambling to get rice or scrambling to get toilet paper because they knew it wasn't coming, right? So networks are really everywhere. The question is this biologically relevant? Are networks applicable to biology as well? And the answer to that is yes. Just like networks are everywhere in our social and our daily lives, they're also everywhere within biology. We have networks within our cells. Our cells communicate with each other and they send signals, and there's a very large cell-cell communication network. We have molecular networks where proteins talk to each other and initiate other proteins, which talk to other proteins and turn other things on. And there's a whole step of how different proteins communicate with each other. An easy one to think of also is the nervous system, right? You touch something hot and your brain instantly knows that you've touched something hot and moves your finger away. That's because there's a network of nerves that go from your finger to your brain, and that signal is sent almost instantaneously. Another one that I mentioned before are social networks, which are also biological in background, in that people interact with each other, and how your network expands or contracts is based on these social networks that exist, right? So why might we want to represent biology in networks? So networks are very powerful tools. And the number one thing that they do is reduce complexity, right? So if you have a list of genes that you've had from a pull down, for example, or you have a list of proteins that interact with other proteins. Looking at that list is very, very difficult, right? You can scan it, right, to look for things, but you're not necessarily going to find any trends by looking at that list. Yeah, you might see a name pop up over and over and over again, but representing that as a picture or as a network would translate that information into themes and into ideas a lot easier. So what I have pictured right here is a SARS-CoV-2 pull-down experiment from a few years ago, from 2020. And what you're seeing here is the results of this pull-down experiment. The red nose are SARS-CoV-2 viral proteins that were used for this pull-down. And any edge is a protein that we found that it interacts with. Now, what you can see right off the bat is you have particular SARS-CoV-2 proteins that tend to interact with more proteins than others. So you can define those as hubs in this kind of subset of the network. Also highlighted in this network are, if you see any areas that are highlighted in yellow, those are actually parts of protein complexes. So you can have an individual protein that's actually interacting with the whole complex. And you've basically taken your list of information and you've translated it into a picture so you can easily tell a little bit better where things are interacting. Another thing that you might not notice in this picture is that you're also integrating additional information, right? So everything red is a SARS-CoV-2 viral protein, and anything in gray is just a human protein, and anything in orange is actually defined as a drug target, right? So it might be potentially something you want to look into for downstream analysis if you're looking to devise or develop a drug that hopefully stops the interaction of the SARS-CoV-2 virus getting into certain parts of the body or getting into your cells, right? So we've overlaid additional information that might not have been part of the experiment. So networks are a great way of integrating different data types. And also, last but not least, the visualization itself allows us to uncover many interesting facts about the data that we would not necessarily have seen if we're just looking at a table of results. So, why would we use network visualization for biological data? There's a lot of different reasons. So the first things we want to do is represent the relationships of the biological molecules, right? So they could be a physical interaction, like with a yeast-2 hybrid or a pull-down experiment where you're actually postulating that two proteins potentially interact. They could also represent regulatory interactions, genetic interactions, or functional interactions. How you define that interaction is up to you and also depends on the data that you're looking at. It is very useful in discovering relationships in large data sets. It's better than looking at your Excel table, right? You're not going to necessarily see an individual protein interacting with, let's say, 20 or 40 genes. If you're just looking through a table, you might notice something comes up a lot, but you're not going to notice, let's say, a cluster of genes from looking through the list. Maybe of your 20 proteins, they all interact with each other, creating this like highly interconnected subsection of your network. So it's also good for visualizing multiple data types together. You don't necessarily have to limit yourself to just the experimental data that you're looking at. It's very easy to pull in information. As an example, in the previous network, we pulled in genes or proteins that were potential drug targets. There were also different data types in there. There was both the SARS proteins as well as the human proteins. So you can pull in multiple types of data into your network a lot easier. You could do that in an Excel spreadsheet as well, but it's a lot easier when you're associating this with some sort of visual attribute that you can easily find commonalities within your network. And on top of that, we can also do something called network analysis. So there's a lot of tools that we can then use with our network to find trends or to find interesting information in the network. For example, you can find subnetworks, which could be highly interconnected areas in the network that could represent potentially protein complexes, for example. You can also look for certain genes that are highly connected or hub genes, right? Does a mutation or does a perturbation in an individual gene cause the whole network to fall apart? You can also look for specific paths within the network, how two different proteins might connect, or different motifs that come up over and over within the network, like a self-regulatory network kind of thing, or a self-regulatory motif. So a few other applications, I guess, for what we can do with these networks, right? So I mentioned this briefly, right? So we can, there's the concept of detection of protein complexes, right? So given your network of, let's say, protein-protein interactions that you've pulled from a yeast-2 hybrid, or even a protein pull-down experiment, you could then look for areas of the network that are highly interconnected and potentially could represent protein complexes. And this is something that's being done in a lot of different species, and is a lot easier to do using networks. Another interesting example of what we can use with networks is gene function prediction. If you have a set of networks, doesn't necessarily have to be protein-protein interaction networks. It also can be gene regulatory networks, or functional networks. You can, if you all of a sudden have a new gene that potentially interacts with a gene of known function, you can use the information that you have to guess or predict a function for this new gene. So gene function prediction is another application for networks that we'll see used. And there are a few other tools that we can use with our networks, including subnetwork-based diagnosis. So we can look for biomarkers, or panels of biomarkers. So a bunch of genes that might be differentially regulated, but also regulated together, and they could form a panel that we can use to delineate as a biomarker or something that's activated in disease. Another interesting thing we can do with networks is between species, we can do network alignment and comparisons. So we can analyze what genes are present across different species, and potentially find pathways or areas and motifs in the network that are evolutionary conserved or not conserved, and how things have changed. And so let's go over just some network basics, right? So what is a network? A network consists of nodes and edges. The nodes is, the node can be defined however you see fit. It does not necessarily have to be a gene, doesn't necessarily have to be a protein. It is something that is connected. It has an interaction or relationship with something else. Now, the reason why I say right now that it doesn't, it's how we define it, because we're going to be using networks a little bit differently than the traditional usage of networks. My guess is that most people have seen the concept of where you have a gene and it interacts with another gene, or you have a protein and it interacts with another protein, or you have a gene that interacts with a transcript, or a drug that interacts with a given with a given other protein. Right? But the basics of the networks is it consists of nodes and edges, right? So it consists of an entity, and it consists of a relationship with those other entities. And you can define that relationship as ever you see fit, right? So when you have a genetic interaction network, each node is a gene, and it's the genetic interaction that is the relationship between those two genes. Right? And so the most common one is a protein-protein interaction, where each node is a protein and it interacts with another protein. And it's actually representing a physical interaction that we think exists in the cell. A genetic interaction is not necessarily a physical interaction of genes within the cell. It's saying that these two genes are related to each other. The same idea with the co-expression network. Just because you find two genes that are co-expressed, it does not mean that they interact physically within the cell. But there are co-expression networks. There's a lot of expression data where where you create these networks where anything that is expressed in the same location can become a network. And it's more relating a different type of data onto a network. It's not the same as a protein-protein interaction, but it's still a network that you can use, and one that is used by a lot of different programs. Specifically, Gene Mania, we'll see a little bit later, they have calculated co-expression networks. So what are additional features, right? We know that we have an individual node and it interacts with other nodes through some sort of relationship. Once we have created our network, there's a lot of different other features we can now define our network by. Specifically, there are topological features, right? So these are features that describe the network. And basically, you can say, well, it's the number of nodes and the number of edges that you have in a network. But it also is, for each node, you can define a degree. And a degree represents how many other nodes this node is connected to. So if you look at a particular node in your network, this one over here, you would just count the number of edges, leaving this node to all of its neighbors in order to get its degree. And now, why is this important? Because, I mean, it brings us to other aspects of the network, right? So you can define something as a hub. A hub is something that is highly interconnected, meaning it has a lot of nodes that it's connected to. This network I have here, this example, is actually highly interconnected. I mean, there is a small subset over here that probably have a degree of 1. They're only connected to this one node, mkRNA protein over here. But there is a lot of connectivity within this network. So there are a lot of different topological features that you can calculate. If you're looking for a specific protein that is highly interconnected, you can calculate the degree of all of the nodes in your network and find which one has the highest degree, for example. Another thing that you can do, you can analyze with these types of networks, is you can also find clusters within the network, so nodes that tend to be highly interconnected, whether it represents a protein complex or whether it represents a common pathway theme, as we'll see later. You can also integrate additional types of data. So here what you can see is not only are there nodes and edges, the nodes have different colors and the nodes have different shapes. And depending on what their shape is and what their color is, means that they're a different thing. So blue squares in this network are transcription factors. Yellow circle nodes are apoptosis genes. And the purple square is the mkRN ribonucleotide protein. What you'll also notice is there's different types of edges, right? So there's blue edges and there are green edges. A green edge is a transcription factor binding to a gene at a promoter, but a blue edge is actually a protein interacting with an mRNA. So there's a lot of information that is stored in this tiny, small little network, right? And these are just a few of the things that we can do when we're analyzing a network. But we can actually go one step further. Visualization. So visualization is actually key, because a lot of times you'll look at a paper and there's just like a massive hairball of a network and it doesn't actually show anything. I feel like more and more it's becoming less the case. People don't just show you a hairball. A hairball is what you see here in A, where you can see a few little colors, but there's not a lot of information that you can get out of this picture. And the way you visualize a network is very, very important. So this visualization example is showing mass spectrometry analysis that generated 400 protein-protein interactions in the pneumonia microbe. And in the first picture over here, you can see this is the whole data set. How we lay out this network is going to be very informative to the person who's looking at this figure, right? So if we then take this network and we just take a subnetwork, right? So then we take a subnetwork with specific functional annotations, which you see in B. Now it's a little bit easier to see individual proteins. You can also see individual colors, and the colors are mapped to different types of proteins, right? As we've said before. But again, how you lay this network out is going to be very informative, right? So now if we take this network B and now lay them out by color, right, which actually has to do with the protein complex that they're part of, what you can see now is a lot clearer of a picture, right? We've highlighted members of the particular complexes that we have in question and their interactions, and we've actually put a circle around that complex. And you could go one step further, as you see in D, and reduce the complexity even more and collapse those complexes if they're not important to what we're trying to say. Like, for example, if we're trying to highlight potential new interactions for each one of these three complexes, you don't necessarily need to see the entire specifics of that complex. And you can actually just generate this type of figure where it's clear for the reader to see all of the new interactions you're trying to highlight. So another really cool thing we can do with network is visualize the network, and how we visualize really determines what the person who's viewing the network is going to see. Okay, so one thing important, what is missing with these networks, and that is dynamics. So when we look at these networks, they are showing a lot of information, but they're not showing the flow of information, meaning we don't know at what point in time this network exists, right? So there's no representation, mathematical representation. There's no stoichiometric modeling or kinetic modeling. That actually is a different layer. It's an additional wealth of information that we would have to add to this type of network. And another thing that's also very important that we're not outlining here is context. We don't talk about cell type. We don't talk about development stage. We're looking at a pinpoint in time of a network of genes and of proteins. Okay, so what have we learned? Networks are very useful for seeing relationships in large data sets. It's important to understand what a node and an edge means, a node being something that interacts, or it's the entity in the network, and the edge is the relationship that those nodes have with other nodes. It's important to define the biological question, know what you want to do with your gene list or your network, know what you're defining. And if you're clear about what you're defining, then your picture makes a lot more sense. And there are a lot of different methods available for analyzing networks. Now, I've just done a brief overview over here. We're going to go over some of those, or a lot of those, in future lectures.